Hi everyone. Imagine this, you are solving a jigsaw puzzle, and just as you are about to complete it, someone mixes in random pieces from another puzzle. Frustrating, right? That's exactly what happens when DNA evidence gets contaminated. Welcome to this lecture on the importance of contamination prevention in DNA collection. In forensic investigations, DNA is like the truth teller. It can identify suspects, link people to crime scenes, and even solve cold cases, but only if it is pure, untampered, and accurately collected. A single drop of sweat, a fingerprint, or even a careless cough can introduce foreign DNA into a sample, and that could mislead an entire investigation. So, today we will explore why preventing contamination is not just important, it's absolutely critical to making sure justice is served. What is contamination? So, what exactly is contamination when we talk about DNA evidence? In simple terms, contamination refers to the unintentional introduction of foreign DNA into a biological sample. The foreign DNA can come from people, objects, or even other evidence samples, and it can mix with the original DNA, leading to inaccurate or misleading results. Contamination can occur at several stages of the forensic process. It might happen during collection if the investigator touches the sample with bare hands or uses unsterilized tools. It can also occur during packaging if multiple samples are placed together without proper separation. Sometimes contamination takes place in transport if the evidence is not sealed properly or even in the lab while the sample is being analyzed. The biggest problem with contamination is that it affects the integrity of the evidence. It becomes hard to tell which DNA belongs to the suspect, the victim, or someone else entirely. This can compromise the entire investigation and may even lead to wrongful conclusions. So, in forensic science, where accuracy is everything, contamination can mean the difference between justice and injustice. That's why strict protocols and trained professionals are essential at every step to make sure that only the original, uncontaminated DNA is analyzed and the results truly reflect what happened at the crime scene. Understanding contamination is the first step in preventing it, and that's what we will dive deeper into next. Sources of contamination. Now that we know what contamination is, let's take a closer look at where it actually comes from, because understanding the sources is key to preventing it. One of the most common sources is the people handling the evidence, like investigators, paramedics, or lab technicians. Even with the best intentions, they can accidentally leave behind skin cells, saliva droplets, or hair simply by talking, coughing, or touching a surface without gloves. Remember, our bodies constantly shed cells, and those tiny traces can carry DNA that easily contaminates a crime scene sample. Next up are poorly cleaned tools and surfaces. If swabs, scissors, tweezers, or even lab benches are not sterilized properly between uses, DNA from a previous sample can transfer into the next one. It's like using the same spoon to taste two different dishes. You can't be sure what flavor really belongs where. Then there is a cross contamination between samples. This happens when multiple pieces of evidence are packaged or processed too close together. For example, if a suspect's shirt and the victim's clothing are placed in the same bag or touched with the same gloves, the DNA might mix and suddenly it's unclear who was there where or what really happened. Lastly, let's not forget about environmental exposure. Dust, moisture, and even air can carry biological material. If evidence is not sealed and stored properly, it becomes vulnerable to contamination from the surrounding, especially in busy crime scenes or crowded labs. So, you see, contamination is not always dramatic or obvious. It can sneak in through the tiniest mistakes. That's why forensic teams must follow strict protocols, ensuring every step from collection to analysis is done with extreme care. Why prevention matters. Why does preventing contamination in DNA collection matter so much? First, it avoids false DNA profiles. Contaminated samples can introduce foreign DNA, leading to inaccurate results that may implicate innocent individuals or exonerate the guilty. Second, it ensures accurate identification of individuals. DNA evidence is pivotal in linking suspect to crime scenes, and any contamination can compromise this connection. Third, it protects the credibility of forensic results. The integrity of forensic science relies on the trustworthiness of its methods and findings. Contamination undermine this trust. Lastly, it's crucial for fair trials and justice. The legal system depends on reliable evidence to make informed decisions. Contaminated DNA can lead to miscarriages of justice affecting lives profoundly. In essence, meticulous contamination prevention upholds the integrity of forensic science and the justice it serves. Prevention measures. To ensure the integrity of DNA evidence, it's crucial to implement stringent contamination prevention measures. Let's explore the key practices. Personal protective equipment. Wearing appropriate personal protective equipment is the first line of defense against contamination. This includes gloves, masks, disposal gowns, hairnets, and shoe covers. These barriers prevent the transfer of skin cells, saliva, or other biological materials from personal to the evidence. It's essential to change gloves frequently, especially between handling different items or after touching potentially contaminated surfaces. Use of sterilized tools and single use collection kits. All tools used in evidence collection must be either sterilized before use or be single use disposables. This includes swabs, tweezers, and containers. Using sterilized or disposable tools minimize the risk of transferring DNA from previous samples to new ones. Proper packaging and labeling. Each piece of evidence should be packaged separately in clean, appropriate containers. Prefer paper bags for biological samples to allow moisture to escape and prevent mold growth. Proper labeling with detailed information ensures that each item can be accurately tracked and prevents mix ups. Maintaining chain of custody documentation. A meticulous chain of custody must be maintained for all evidence. This involves documenting every individual who handled the evidence along with the date, time, and purpose of each transfer. Maintaining this record ensures the evidence integrity and admissibility in court. By adhering to these prevention measures, forensic professionals can significantly reduce the risk of contamination, ensuring that DNA evidence remains reliable and credible throughout the investigative and judicial processes. Legal and ethical importance. Let's dive into the legal and ethical significance of preventing contamination in DNA evidence. Legally contaminated DNA evidence can be deemed inadmissible in court. Courts require the evidence be collected, handled, and analyzed following strict protocols to ensure its integrity. If contamination is detected, it undermines the reliability of the evidence, potentially leading to its exclusion from the trial. This exclusion can weaken the prosecution case or hinder the defense's ability to prove innocence. In some instances, contaminated evidence has contributed to miscarriages of justice, where innocent individuals were wrongfully convicted or guilty parties were acquitted. Ethically, forensic professionals have a duty to uphold the highest standard of practice contaminated evidence, not only jeopardize individual cases, but also erodes public trust in the forensic science system. Maintaining the integrity of DNA evidence is crucial to ensure that justice is served fairly and accurately. Proper handling and analysis of DNA samples reinforce the credibility of forensic findings and support the ethical obligation to prevent harm caused by erroneous evidence. In summary, preventing contamination in DNA evidence is not just a procedural necessity, it's legal imperative and an ethical responsibility. By adhering to stringent protocols and maintaining the highest standards, we safeguard the justice system's integrity and uphold the public's confidence in forensic science. Thank you for joining me on this journey through the vital aspects of DNA evidence and the paramount importance of preventing contamination. If you found this lecture insightful, please consider liking the video and subscribing to the channel for more content on forensic science and related topics. Your support helps in disseminating knowledge and fostering informed discussions. Stay curious, stay informed, and until next time, keep exploring the fascinating world of science. Bye bye.